Hey guys, so this particular lecture is all about carbon and how it constitutes the building blocks for life. So let's do it. Yay. Why do we care? Well, all life is built on carbon. I know we used to think that all life was built on water, and water does play a very important part in biotic life. However, all life is really built, like physically, literally built on carbon. So when you think of a cell, we know that a cell is about 72% water. What's the rest of it? Well, after that 72%, about 25% of what's left is going to be some form of a carbon compound. And they come in four types: carbohydrates, lipids, proteins, and nucleic acids. That still leaves us with about 3%. That's mostly salts like sodium and potassium and chlorine and stuff like that. So 25% is a big chunk. That's why it's important. That's why we study it. And that's what organic chemistry and to an extent biochemistry really does focus on. So here we go. So the study of carbon is called organic chemistry. If you remember me telling you, it wasn't exactly my favorite subject in college, but it kind of grew on me towards the end. So here's what you have to think about. Carbon is a very cool little atom. It doesn't look like a whole lot, but it has some very unique and special properties and abilities. The first of which being its bonding properties. The way it can connect and form bonds are unimaginable. You can't really get that in any other kind of atom. The other thing that you have to consider is the fact that it forms very, very stable covalent bonds. And when we're talking about building life, I would think that being stable and being strong would be two very important things on the list. So carbon kind of fits the criteria all the way around. So here's the part that killed me in organic chemistry. When my professor would draw things on the board, it would look like this. Oh, that's easy. It's a C in the middle, and it has four H's. I mean, CH. Hydrocarbons were even like my initials, Chandra Henry, CH, I was like, oh, this is going to be great. And then he'd draw some other things, and they'd look like this, and I'm like, oh, still good. Then he'd draw stuff that looks like that, we're still good. But in actuality, these molecules aren't flat. So this is a simplistic representation of something that actually works in 3D. This is what the actual molecule looks like. And if you couldn't relate these two, when you got to some of the harder things, especially some of the reactions, you're lost. That's why I was lost, because I'm female. Our brains don't operate in this kind of mechanism. So remember I told you I literally bought myself a kit and would make these? Yeah, I would sit in the library with Tinker Toys pretty much and make models. And people probably thought I was crazy, but at the end of the day I got an A, so who cares? So that's one of the things that you're going to have to wrap your mind around. Carbon forms molecules that are really, really complex. They look flat when you put them on paper, but in actuality they're 3D models. They stick out at all kinds of angles. So, yeah, it gets interesting. Okay, so let's talk about what a hydrocarbon is, because you're going to start hearing this word a lot. A hydrocarbon is a combination of carbon and hydrogen molecules. It is nonpolar. So because it's nonpolar, it's not soluble in water, which is really good. You don't want your building blocks to break down in water, because when you think about the fact that they're found in cells, and you think about the fact that cells are 72% water, if they were polar, that would create a really big issue. Hydrocarbons are hydrophobic. They actually don't like water at all. They're very, very stable, and there's very little attraction between the molecules. So what that means is this molecule isn't pulling on electrons any more than this molecule is versus this molecule. They each hold equal attraction for each other. And because of there's so little attraction, they tend to be a gas at room temperature. So they can grow. They can also shrink, but we like it when they grow. And it's really simple. You're just adding on some units. You throw in an extra carbon, and that carbon can bond a certain number of hydrogens. And they don't only grow linearly, like right here you're seeing them grow linearly, but here we can also make them branch. We can change their shape. We can create ring structures like they show here, and we can throw in double bonds and maybe even triple bonds, and that changes how things fit together. So you're looking at a molecule that is highly versatile. When you think of the fact that life is so diverse, being a versatile building block is a really, really good thing. Another good thing on the praise list for carbon is the fact that it forms isomers. Hopefully you remember what that is from chemistry, but in case you don't, it's when molecules have the same molecular formula, but they have different structures. So they're put together differently. They're assembled differently. And this is kind of my mantra all year long, so get used to hearing this a lot. But in biology, chemical properties are going to affect shapes. Shapes are going to affect function. Okay, so function relies on shape and shape relies on function. They go both ways. So if I can mix up my structure, it means that I can do lots of different roles or functions within wherever it is I work. That's another really useful thing. So here's an example of how form can affect function. Your body naturally creates an amino acid called alanine. Alanine comes in two forms, what we call an L form and a D form. The proteins in your body make L-alanine, not D. It just works better structurally for us. So for medications that either help with the making of proteins or give us additional alanine, they need to be L version activated and not D version activated. If those medicines get switched up, we can have an issue. So these are what we call stereoisomers. They're mirror images of each other, in other words. So let me show you how it can get real sticky real fast. In the 1950s and 60s, a drug called thalidomide was prescribed to pregnant women. It helped to reduce morning sickness, so everyone felt better. But the issue was it was a stereoisomer for the same alanine that we were talking about. The results were some very severe birth defects. If you look at these children, like in all these pictures, you notice that they look fairly normal except for, their arms. Look at how small and reduced their arms are. That was one of the birth defects that thalidomide caused, and it's all because the wrong version was activated. The D version was activated as opposed to the L version. So form affects function. Highly important. So because carbon is so diverse, it doesn't have to just bond with hydrogens. It can bond with a couple of other types of atoms. So it's very easy to substitute other atoms or even whole groups around carbon. So I'll give you an example. Let's look at ethane versus ethanol. So down here, that's the structure for ethane. C2H6. Okay. Ooh, I guess my structure for ethanol has disappeared. I guess I can draw it for you, or try to draw it for you. Don't laugh. So ethanol also has two carbons. The first carbon has 3 hydrogens bonded to it, whereas the last carbon has 2 hydrogens and a hydroxyl group. Okay, so by getting rid, let me write the structural formula if I can. The structural formula would be C2H, hold on. So C2H5OH. So by getting rid of 1 hydrogen and replacing it with 1 hydroxyl group, we've pretty much changed what we're looking at. Ethane is nonpolar. because it's all carbons and hydrogens. Okay, nothing to dissolve, it can't dissolve in water. Ethanol is very polar, and it's polar specifically because of this OH group. That OH group makes it polar. Ethane's a gas, ethanol's a liquid, and the two of them have very vastly different biological effects on humans and other living things. So what's a functional group? A functional group is an organic molecule or parts of organic molecules that are actually involved in chemical reactions. Don't forget, chemical reactions are all about losing and gaining electrons. These functional groups give organic molecules very distinctive properties, sometimes properties that are unique just to that class of macromolecule or organic molecule. Here's another thing you're going to have to get used to. They're called organic molecules. They can be called macromolecules. They can be called biomolecules. Get used to it. You'll hear all three terms interchangeably. So there are three main kinds of functional groups. They are hydroxyls, carbonyls, carboxyls, amino. That should be amines, but it doesn't matter. Sulfhydrides and phosphate groups. What these guys really do, all six of them, is they take hydrocarbons, which are very, very hydrophobic, and make them hydrophilic. By making them hydrophilic, it increases their ability to dissolve in water, so it increases their solubility. So let's look at another example. This is another example of how function and form go hand in hand. We're all familiar with hormones. Male and females both have them. The classic example is males have testosterone and females have estrogen. The correct name for estrogen is estradiol, by the way. If you look at the actual hormone, I'm going to outline that part for you in green. That would be, or in purple, whichever my pen touches on first. That section here, that section there, it's the same molecule. The base structure, the carbon skeleton, is exactly the same for testosterone versus estrogen. What's different are the functional groups and where those functional groups are attached. Okay, so that's where the uniqueness comes into play. Because of the type of functional groups that the different hormones have and also their site of attachment, it makes them react with different targets within the body. Because they're targeting different sections of the body, you're going to have different results at the end of the day. So, form and function, and functional groups. So let's start with the hydroxyls. Hydroxyl groups are OH groups, so an oxygen and a hydrogen. Any compound that has an OH is called an alcohol. So typically the name ends in OL, so methanol, ethanol, butanol, propanol, any kind of ol. And you can attach it anywhere. It doesn't have to be attached to a specific place. As long as it's there, you have a hydroxyl group, you have an alcohol of some type. Now, methanol is not good to drink, but what we drink as alcohol, the drug in alcoholic beverages, would be ethanol. The next group is called a carbonyl, and it's what happens when you have a carbon double bonded to an oxygen. Now this one's tricky because it really does depend on where that double bond is in the molecule. If the double bond between the carbon and the oxygen happens at the end of the molecule, so please note that end could mean on the right, but it could also mean on the left, you have an aldehyde. So if it's on the extreme right or the extreme left, this is not just a carbonyl anymore, it's called an aldehyde. If the double bond between the carbon and the oxygen happens in the middle of the molecule, like it does here, now we have a ketone. So we call them different names based on the location of the double bond between the carbon and the oxygen. So one more time. If it is found, if the carbon double bonded to the oxygen is found on the end of the molecule, that could be the left end or the right end, this is called an aldehyde. If it is found in the middle, so right in the middle of the molecule, it is called a ketone. Okay, so if you're familiar with acetone, that's an example of a ketone. The next group is the carboxyls group. These are acids. They're characterized with a COOH, so carbon bonded, double bonded, to an oxygen and single bonded to a hydroxyl group. This is what it looks like up here. Compounds with this functional group are all acids, and the most important ones to us biologists would be our fatty acids, which make up our lipids, and our amino acids, which make up our proteins. Okay. Another term, the correct acid term for this is actually carboxylic acids, but yeah, you get the point. Okay, now we're on our amino groups. So this one's pretty special to just this category. Your amino is going to be characterized by a nitrogen single bonded to two individual hydrogens, and compounds with this arrangement are called amines. Specifically, we're talking about amino acids, so these guys are found in proteins. The amino group tends to act as a base, and they have the ability to pick up hydrogens from solutions. So that NH2 is technically ammonia. It's the base root for ammonia, and those have the ability to pick up hydrogens from solution. Okay, sulfhydride is the next one. It is a sulfur atom bonded to a hydrogen atom. These are called thiols. So any compound that has that sulfur bonded to a hydrogen, is called a thiol. What they tend to do is they're actually stabilizing forces within the structure of proteins. So they add on some additional strength to some of those weaker bonds that you will soon realize actually hold proteins together. Okay, phosphate group. This one you should be familiar with from freshman biology. Big structure within DNA. Without this guy, DNA kind of wouldn't really work the way that we need it to. Also found in ATP, the energy molecule of the cell, so really important structure. Phosphate groups are obviously phosphorus, bonded to 4 oxygens. 3 of them are single bonds, and 1 of them would be double bonds. They are connected to the carbon through one of the oxygens. Now, because you have all of these oxygens, you have a ton of negative charge, okay, which makes this molecule very, very reactive, which is why we find it in ATP, because it's an easy way to transfer energy between organic molecules. All right, so now we're going to move on to looking at those 4 macromolecules in a little bit more depth. Here we go. So like I said, another name for these are biomolecules. So get used to seeing both terms, macromolecules, biomolecules, same thing. And really what they are, they're small organic molecules joined together to form a large molecule. So hence the name macro, because macro means big. So one way to think of them is as big molecules, but another way to think of them is as life molecules, hence the name macromolecules versus biomolecules. Like we mentioned before, there are 4 classes, and they are carbohydrates or carbs, lipids or fats, proteins, and nucleic acids. And together the 4 of these are the building blocks for every living thing. Alright guys, sorry about the picture. I know you're seeing refraction lines and stuff, but when I imported my PowerPoint, the little the little images like these things here, they didn't show up and I wanted you to be able to see those, so I just took a picture of my screen. So here we go. Polymers. Poly means many. So pretty much what a polymer is, it's a long molecule made by repeating units that have all been linked together in the form of a chain. So we take the same thing and we just make millions and millions of copies of it, and then we smush it all together so that it stays. I always like to think of like links in a chain or like Lego pieces, because they all kind of look the same and you just build them one on top of each other and now you have like a giant Lego wall or whatever. So the little units, the repeating units, are called monomers. They're the building blocks. They're the things that make the polymer. So a monomer would be like one link in a chain, and the polymer would be the entire chain. You want your macromolecules to stay together. So it would make sense that the strongest bond that we have would be the bond holding these things together, the bond that's holding these monomers together. So the bonds that hold polymers together are covalent bonds. Well, how do we make them? Through a process called dehydration synthesis. So synthesis means to make. Dehydrate means to dry out or to remove water. That's literally what you're doing. You are taking the water out of the equation. And when you remove the hydroxyl group over here, excuse me, and the hydroxide group over here, these two ends can now bond together because you've left some electrons there that are attracted to each other. Hence the formation of a bond. So dehydration synthesis or condensation is how we make polymers. The other term, like I just said, is condensation. You might see both, so you need to know both. Polymers are made by dehydration synthesis or condensation. I hope the green shows up. It's looking kind of wonky. Let's try the purple. Let's try the red. Condensation. Okay, so dehydration synthesis is the same thing as condensation. You're just removing water and allowing a bond to form in between where those two molecules were. Okay, same situation. Had to take a picture. So, like I said, how do you build the polymer? You do it by taking the water out. You have to open up bonding. You have to open up space so that a bond can be created. The way we get the water is one of the monomers is going to donate the OH group. So like this one here is giving its OH group away. The other one is going to donate the hydroxide group, And water is H2O, so we have two H's and an O. H2O, water. Now, it doesn't happen spontaneously. It's not like you just click your fingers and water gets pulled off. It requires energy for these things to happen. Excuse me. And it also requires enzymes for this reaction to happen. And again, the reaction is called dehydration synthesis or the condensation reaction. It is how polymers are formed, how they're made. Okay, so if building a polymer requires you to take out water, then the opposite is true to destroying a polymer or breaking up a polymer. And that reaction is called digestion or hydrolysis. So lyse means to break and hydro means water. So literally water breaking. You're going to add water back into the polymer and it's going to cleave off your H and your OH and they're going to reattach to the ends. Again, this requires enzymes and it requires energy. So literally as you're adding this water, one of those H's, so the H plus, the hydroxide, is kind of waiting to attach to this molecule right here. And the OH minus, or the hydroxyl group, it's waiting to attach to this molecule right here. So when the enzyme breaks the bond and creates that space, there goes your hydrogen and there goes your OH. And now we have the monomer being cleaved off or being broken off. All right, that's it for our introduction to carbon and our introduction to carbon and our introduction to macromolecules. So as usual, come ready with your questions. See you next time.